Hello?
Hello.
How are you?
Oh, I'm good. I just don't have I haven't plugged in my camera because my camera tends to knock my sound out.
Oh, okay. Yeah, you were telling me that before.
Okay. Well, so I'll just sort of sit here and watch and yeah, I don't know.
All right. Ujjwal, how are you?
Hello, am I audible?
Yeah, you're audible.
Hello, guys.
Hi, how are you?
Fine. How are you?
Okay. Hello, Tom. Haven't seen you in a
Hey.
while.
Yeah, I know. Yeah. Just thought I'd check in.
Yeah.
I gotta leave at 9:30, but you know, I gotta go teach a class. But yeah.
Okay, yeah.
Yeah.
Hello, Mayukh. And Maniak. Hello, Jesse.
Morning.
Okay. So why don't we get started? Welcome to the meeting. We have our GSoC final presentations today, and then we'll talk about some of the things that might come after GSoC with respect to publishing something on this, and then a couple other things after that. So Ujjwal and Mayukh told me that they had their presentations ready to go, so who wants to start first?
Yeah, I could go first.
Okay.
Can you hear me?
It's very
Am I
low.
audible?
The volume is very low.
It's very low. Yeah, is it better now?
Yeah, it's better.
Is it better now?
Yes.
Yeah. Okay, fine. I'll just start then.
Okay.
So let me just share my screen.
Okay.
So can you see it?
Yes.
Yeah, so I'll just can you see it now?
Yes.
Wait,
It's coming up, but yeah, I can see
yes. Can you see it?
Yeah.
It's full screen, right?
Yes.
Yeah, okay. So then I'll start. So hello everyone. So GSoC has come to an end after like 3 long months. Not exactly long, but still, sometimes it was. So community bonding. This is just basically like a brief look at the developments and the deviations in the last 3 months. So yeah, first is community bonding in phase 1. So initially I just started out with some simple stuff just to dig around and see what DevoLearn and what DevoWorm was all about. So while I was in the community, I also like built this computer vision pipeline which could track worms, like it could track multiple worms from like a video and all. So this was just a computer vision pipeline. So I actually developed another one of them. This one could skeletonize the worm and save save the positional data as CSV files. So this is also up there. These were not very important later on, but they're like just interesting, and these are just exploratory work. So moving on. So then I started out by I just developed an Lstm model which could like predict the curvature of the worm's body from the time series data containing the distance between the head and the tail. Basically, the curvature is inversely proportional to the distance, right? For a given like normalized coordinates, like for a given normalized coordinates like set, this worked. So, but this was even like it was not perfectly accurate, and it was also part of like the explorations that I did. So then moving on. So then what I did was principal component analysis on spatial data of embryo cells. So this basically helps us visualize like how cells descending from the common ancestors had common like parameters. Like I did principal component analysis on like 4 parameters. Those were like position, XYZ, and time. and then I brought them down into 2 dimensions, x and y. So I plotted them. So for each lineage, as you can see on the graph to the right, can you see the graph? Is it large enough?
Yes. Yeah, I can see.
Like,
Yeah,
you see
enough
it?
probably. You might zoom in a little
Yeah.
bit
So
if you're.
you might be able to see it. Yeah, I don't think zooming in is possible. I'll share the slides after this.
Okay.
So what it shows is that basically, like the lineages which have common ancestors lie on like similar positions within the principal component space. This is what it basically showed. Like, this is an interesting visualization. Like, things could be done on this in the future. So then pictures of from the worm image dataset. This, like, I found it like near the end of phase 1. So then I started working, and my prime goal was not exactly this dataset, but to build a pipeline for segmenting other stuff later on. So I actually kind of recycled this pipeline later on for another model, which I'll be showing soon. So then it's phase 2. Yeah. So moving on. Then extracting metadata from embryo time lapses. Like, we trained the ResNet 18 architecture to determine the population of cells of various lineages within the C. elegans embryo. All the videos were from the EPIC dataset, and the model was trained, like, it was accurate until 250. After that, it was deviating. So we just kept it at 250 cell stage, around 250. Mostly it was like 249 to 48. So on the graph, you can see the graph, right? I guess this is big enough. So in this graph, the dashed lines are the predictions made by a deep learning model, or someone who actually manually annotated the EPIC dataset. So it's pretty close, I guess, and it could give these kinds of results consistently on different types of videos or different types of photos. So this worked out pretty well. So then this is another visualization of what's actually going on in the graph. Like, this graph, a more dynamic version of this one would be this one. So as you can see on the left, you can see the cell slowly, like, I mean the embryo slowly, like, multiplying its cells. So it's like basically undergoing the process of embryogenesis. And to the right, you can see the predictions made by the deep learning model, like the dashed lines. That is, like, if you just consider the A lineage, then it's the orange dashed lines versus the blue dashed lines. They're pretty close. So, like, and it can do kind of like live predictions from videos and stuff, which we'll come to later on. So moving on, the next thing that I did was, like, it was kind of a tangent again. So what it did was, like, I made a GAN which could generate synthetic images of the embryo. Like, but we can, like, develop this more later on. Maybe we could develop a conditional GAN. What it could do is that we would give it populations, and it would give me a synthetic image of an embryo which has approximately those cell populations within that image. So yeah, until now this is not a conditional GAN right now. So it basically returns like images of random populations within the embryo. So yeah, moving on. Can you see this GIF? Is it loading properly?
Yeah, it looks. I mean, I can see the GIF.
Yeah.
Little,
okay, moving on then.
Yeah.
So then, deep segmentation model to segment the C. elegans embryo. This segmentation model was actually later used in DevoLearn to build, like, an easy to use segmentation tool. We'll come to that soon. But as you can see, like, this video is like a pretty, like, noisy kind of a video, and then it could segment, like, it pretty well. Like, I think it got an IOU score of above 85 to 90%, I guess. I don't know the exact, like, latest figures. And then it could also extract the centroids. So then we actually developed on this, like, soon after this, which I'll be coming to. So, and in fact, all of these models are trained on noisy images as well, so that they can handle, like, really bad quality or noisy, like, images from different types of sources. So moving on then, phase 3. So say hello to DevoLearn. DevoLearn is a Python library which can help accelerate data-driven research on C. elegans embryo by automating the process of collecting metadata. So what it does is basically it's kind of a culmination of all the good stuff that I made during phase 1 and phase 2, and we just integrated some of those models, not all, to form a very simple to use kind of a Python library which is already there on PyPy. Like, people can easily install it and use it, like, to automate the process of collecting metadata. Like, I could give this example. Like, DevoLearn could one day help fuel various like data-driven research like approaches. Like I'll show this GIF to the left. Like, as you can see, it can extract like the centroids from videos, and then it can like save the use to visualize things like these. Where what I did was that like even though I'm not good at developmental biology, like I could just look at the mathematical aspect of it. So I just plotted a distance matrix which could like visualize the distance between each centroid of each cell. So these are the kinds of interesting things that you can do. And on the right, you can see another GIF, which is basically like an approximate map of the centroids of the cells within a C. elegans embryo from a given instance of its embryogenesis process. Why approximate? Because it's like averaged out. So then like if I move on to finer planes, then it would give me better data, like better centroid data. So then moving on. So this is basically the structure of how DevoLearn is right now. There are three main models. One is a lineage population model. This lineage population model is basically this model, which can like take in images and videos, and it can return like CSV files containing the populations of each frame. Like frame by frame, it can return in rows. It can even save it. Like it's almost like pretty much automated. So that was the lineage population model. And then the embryo generator model is the GAN that I talked about. And then the embryo segmenter is this one, of course. Like this is also integrated into DevoLearn, which like as little as 3 to 4 or maybe 5 lines of code, you can like use each one of these models for like, and you can also use them on like videos and images. Like it is compatible with both. And the thing is that the person who is installing this library doesn't have to necessarily install all of the three models. Like whenever a person calls one of the models, only then is the model file downloaded, which is like 50 MB each. So yeah, moving on then. Looking ahead. With the right contributors, DevoLearn could one day become a useful tool. So these are basically the things that could be improved in DevoLearn, which I've thought about lately. Some of them are also on the GitHub issues. You could check them out. So the contributors could help by improving the model's accuracies. They can submit more deep learning models which could be added if they are in alignment with our interests, like on the C. elegans or embryogenesis itself, and if they are accurate enough. And then DevoLearn currently works with only images and video files and CSVs, and it's not like it cannot take .tiff or .mat files like as input. So we could extend that support soon enough. Like someone could do that. I'll have to raise an issue on this. And then it currently works on the GPU. It currently works on the CPU. I mean, sorry, but if moved to the GPU, it could like be much faster. So and it won't be a lot of effort to move it to the GPU optionally, because Torch already supports CUDA. And then like one could build a DevoLearn GUI for non-programmers, like some kind of a like an exe file which would open up some kind of a GUI, which would help us reach a much larger user base. And overall, DevoLearn has the potential to be much better, and I'm ready to help anyone who wants to make it better. So yeah, thank you everyone for your help and feedback. I'm looking forward to more exciting stuff. Yeah. So
Well, yeah, it was very good. Thank you. Any questions? So yeah, this is the summer code project for the pre-trained model. So for those of you who don't know, we have the DevoLearn platform, which is DevoLearn, the program, and then we have Ujjwal's, what he's going to present on, which is the revamp of DevoZoo, and then also some other development. So this is the link to DevoLearn in the chat. Mike just put it in. If you want to, the organization is DevoLearn, and then there's a DevoLearn repository underneath that. And you can download his program and work with it. We also should talk about if you want to do anything with the, I don't know how it's integrated into this, but the movement data that you were showing. And what else was there? So that's the quick summary. So he's writing up his GSoC proposal. So anyways, I encourage you to go look at it and play with it if you want, and see if you want to contribute. There are a number of pathways. We're kind of setting up issues on that. I don't know if we have an issue board at the level of I don't know about the organization, but at the level of the repository, we can definitely set up an issue board. So for DevoLearn, the program, we can set up an issue board for that. Yeah, and you can try out DevoLearn in a Google Colab. So Mayukh has made a nice Colab notebook of everything in DevoLearn. So let's move on to Ujjwal and hear from him.
Let me just quickly share my screen.
Okay.
Is my screen visible?
Yes.
Can you see the presentation?
Yes.
Yeah, so this is the not I can say end, but it's the end of GSoC period, and I have enjoyed a lot, and so this is the final presentation that we have to give. So moving forward, sorry. So just a quick good news that attracts me about the Neuralink, if I know most of the people but the main thing I want to highlight about Neuralink is about the size that they are getting. They are making it much more compact, which amazes me. So I think it should be given a little emphasis on it. So we can discuss it about later. So moving on to the presentation. So yeah, hi, I'm Ujjwal. Most of you people know me. I am a third year graduate, undergraduate student at IIIT Delhi. And I am this year selected for the project Open DevoCell integration, and these are my Twitter, GitHub, and LinkedIn links. So if you want to connect, so you can connect. Quickly moving on. So Open DevoCell. So Open DevoCell is the project that it is given to me this year, and I was fortunate enough to do it. So aim of the Open DevoCell is to connect the DevoWorm and OpenWorm ML architecture, and as listed, to improve the web interface and to develop new intuitive and beautiful interfaces. Reorganize DevoZool. DevoZool we are coming in few minutes on it. Open DevoCell tool customization, new front end, and improvisation. So Open DevoCell is something that is being prepared by Vinay in last year, and this year we try to improve it and incorporate much more things in it, and to give a fresh look completely. Devo editor. So this is the thing that we have completed. I have completed. It is not linked yet, but it will be linked this week. So, and this is kind of an editor so that researchers don't have to use and download and label the images manually by installing softwares and doing manual label. They can do all these things online. So this is the kind of thing that we are trying to provide through Devo editor. I will come to it later. And the DevoLearn library and tutorials. So DevoLearn is a library, is a project, Mayukh's project, and he has done an incredible job on it. So I have a very little contribution to it, and more of kind of a tutorial base and helping setting it up. So it is a very good project, I must say. So community bonding period. So community bonding period is one of the phase where we have interacted with many people. So we have bonded with people. We have got opinion from many people on our project, and we have get how to proceed further. Then we have weekly meeting and onboarding. So Brad Lesser and Vinay have onboarded us on the project. They have told us what we have to do and how we have to manage things and all these stuffs in a meeting. And then we have office hours where we have described our project in detail, and we got some few feedbacks from the community to work on, which are very beneficial for us, and we have implemented them as well. So onboarding process was very smooth, I guess, and we have done things very smoothly this summer. So this is about the community period. Moving on. So there is a DevoWorm AI. So DevoWorm AI is basically a DevoWorm group AI center, which we aim to build all of the things on once. So where people can find new resources, reading materials, DevoWorm Academy resources, and other online tools and notebooks. So it is kind of all the things that we have done right till now, and we want to improve it. So as you can see in the GIF, it is the older version of DevoWorm AI. We have edited it and we have made it a little smooth and little less like flashy twice. So we have tried to come up with that things. So this is basically about the DevoWorm AI tool, and it consists, as you can see, all the history of DevoWorm, like the tools for getting started with the neurobiology and embryogenesis process, and like C.Elegans project and DevoWorm Academy, etc. So I will like try to give you a live demo of it in a while. Then we come to DevoZoo. So DevoZoo is the DevoWorm Group data center, which is maintained by DevoWorm Research Group and Orthogonal Research and Education Lab. And they have hosted we have hosted multiple datasets online for the purpose of helping researchers around the world. So like this website already exists. So what I have done this year is like to give it a new fresh look, so that people don't have to click on all the links and like have to wait for like, oh, what is the dataset is. He has to go, and sometimes like it is a drive link, so people have to ask for permission. So like they can readily see on the website like what dataset we are providing to them and what kind of dataset it is. I will like explain it in a bit, like when I will show you a live demo, like how we are trying to segregate it. C.Elegans. So if we can say, let's C.Elegans segmentation or Open DevoCell version 2. So basically, it is a project that Venus has started in the last GSoC year, and like I have improved it this year, and Mayukh have given many good insights in DevoLearn library to improve the segmentation procedure, and we have implemented that in our portal. So as it is written like that, it is inspired from the last year Open DevoCell project. This year we tried to incorporate more things to the existing model and app, and to provide the full details of DevoLearn and C.Elegans on website itself. So it is not just a tool; it is like a resource center where you can come and read about C.Elegans segmentation procedure. You can read about the library that we have built recently. You can try our tools online. You can contact maintainers. So it's a kind of a mini resource center, you can say, under DevoWorm AI. So basically, the aim of DevoWorm AI is to connect all the things that DevoWorm Group has on one portal, like under one roof, so that all the things can be accessed. You don't have to go here, like you have to, if you want to access some data, you have to ask us, like you want that data, and we have to provide you permission. We want to make it as open source as it can be, so that people around the world can very easily access them and can very easily use them, and it can help their research in their research purposes. Then the DevoLearn, as I have said, like it is majorly Mayukh's project, so I have done a little bit contribution on it. So it is a kind of a PyPy package that is deeply, very well explained by Mayukh. So I am not going to spend much time on it. So yeah, it has definitely a very much potential. Like it is right now limited to C.Elegans only, and we are trying to incorporate more and more models in it. And as we get the new contributors, which we are getting, and we are getting very good progress on DevoLearn library and DevoLearn page itself. Like there are many visitors on our packages, and like all these like new contributors are already joining us. So yeah, so this is about DevoLearn. So before like thanks, I would like to just let me see. Yeah. So this is the DevoLearn resource. Can you see the website?
Yes.
So this is the DevoWorm AI Center that we are talking about. So it is like the motto, like the first initiative to create a first virtual organism. So you can come and join us on our journey. So then when you scroll it up, you got C.Elegans. So C.Elegans, like it is explained like what it is and what it is trying to do, what kind of technology it incorporates. And so I will like explain this in a detail in a bit. First of all, I will just go through the website. So after that, there is a digital bacillary notebook that I have posted on GitHub and will be linked to it. So it is same as C.Elegans embryogenesis, just a different diatom. After that, general segmentation model, so we have created some notebooks for people to get onboarded about what image segmentation is. So we have tried to give them as much detail as we can. Like if a person is interested in going deep, like how the image segmentation model is done, and he has some experience in computer science, he can go as like deep we have made it possible, like as we can. He can go and see what the things are really, how the things are really happening in that notebook. So this goes that. This is the DevoZoo and Devo methods that I have explained in my presentation. Like we have revamped the DevoZoo and Devo methods. So Devo methods work is like kind of still pending, and DevoZoo is completed. So let me just quickly show you DevoZoo. So after that, DevoWorm Academy, this is the thing that in which I believe Brad Lisser and Jesse is working on, and I think we will shortly link all the things to this. So as you can see, it is a kind of a skeleton for the DevoWorm group, like where you can add more things and you can link them on. So when the DevoWorm Academy course is formally got ready, we will just link it here. This is about the history of DevoWorm. If someone is interested, like how DevoWorm has started, like what are the things that have been done. So it is a kind of a short timeline that I have got from the meeting, which we can surely update in a bit as the time moves forward. After that, there's a quick numbers about like how DevoWorm group has grown. It is about like the founder and the team of DevoWorm, ML team and advisors, like the main people who are involved in this uh, group. And after that, you can contact us through your like if you have any questions, you have anything that you want to contact and you want to make things right, so you can definitely contact us. If you want to like have some bugs you want to report us, you can do that as well. So there's a kind of a menu here. So moving forward, C.Elegans. So there's a C.Elegans segmentation tool that we are talking about. So as you can see, like it says, like it takes the microscopic images of embryogenesis process, try to segment the cell bodies and calculate the centroids in the area of each cell. You can download these things in a form of CSV file from our portal. So there is a project which do all these things in an unsupervised manner, so researchers don't have to get up and like don't have to get up and like label each and every cell. It is it in an unsupervised manner, so it will make research quite fast. So as you can see, this is the intro. So it is an introduction about cedar elegans and their embryogenesis process. So this is the thing that we have taken from Worm Atlas, which is cited below. So as you can see, like these are the different phases, like fertilization, proliferation, castrulation, elongation, etc. So you can visit Worm Atlas to know more about it. And it has other links like library as DevoLearn. There is a DevoLearn library link where you can get quickly started about like how to install DevoLearn, what are the different functions that you can use, and all these things. So it has, as the Mayukh has explained, like we have tried to keep it as simple as we can, so you can just take a few commands and you can do your thing. You don't have to just go deep into like what are the tools and what are the functions that are being responsible for producing results. You just have to use the library on an abstract level, so that you just know the commands. You just go and type the commands if you are not related to computer science, and you will get your things done. So we have tried to give this thing here. After that, you can try our tool online. So it is a beta version because of the memory restrictions and network restrictions. So it kind of produces a result which is not as good as you can do with the library, but you can get a gist from here, like what our library can produce. So you can just simply upload your files here and upload, and it will produce results like this. So I don't have C.Elegans photo right now in my this laptop. So, but you can definitely upload the photos and upload it, and it will produce a result. I have tested it multiple times. Then the contact information, like if you have any issues or any trouble, like if you are facing, you think like something has to be improved, something can be improved, you can just go and create an issue on our DevoLearn repository, and Brad Lesser, Mayukh, Vinay, we are very active, and we will definitely reply to your query as short as we can. And if you are not familiar with the GitHub, you can reach any of us. All our contact details like Twitter handles and anything are mentioned on GitHub repository itself. So this is a quick tour about C.Elegans segmentation. Then they explore DevoZoo data here. So there is a DevoZoo new interface, like new DevoZoo, if you will, and click on the below button to visit the GitHub repository. So as you can see, you can click and go to DevoWorm repository. So the major thing that we have done this year on this is, if I can show you the previous DevoWorm website, so we have not removed anything from here as of now. Oh, I guess I have renamed it, but nevertheless, I remember the. So yeah, this is the older DevoZoo, which, as you can see, like it has all the things there, right there. It has nothing to be like which has extra there. But like you have to click on certain links to get to know about like what kind of data you are providing. Are you providing microscopy images? Are you trying to provide like uh, Excel files? Are you trying to provide some folders, some text file? So you have to like click on this link, go to like some of them are uploaded in Drive. So you sometimes you have to ask for access and all these things. So what we have done this year is like we have improved the front end as well as we have given the icons like lineage tree is an Excel file, which means like it is in a format of .xlsx or it is a CSV file, like Microsoft Excel file or a CSV file. As we go to there, you can see like if we say at nematode microscopy movies, so these are the 5 major movies that we are providing. So you can just readily see it here by clicking on play, and it will be shown you here. And if you want to, if you are interested, yes, like you want this kind of data set, you can click here, and you will be redirected to the original data set folder where there are multiple videos like this. Then moving forward, like there is a C-square data set, a fruit fly data set, and all these things. So after all these data sets, this is the latest tweets like it is like kind of a extra function like you can get to know like there is an orthogonal lab and OpenWorm existing on Twitter. So if you are very interested, you can follow us there as well. Right from here. And like if this space is right now empty, but if in future we set up a page for DevoLearn, we can easily embed it here. That's why this space is empty. Which I surely believe like if we got a right contributor, we work on a way we are working right now after GSoC as well. It will get a very useful tool. At least I believe it. Then it is basic like our vision and research summary. There are some microscopy tutorials like for the people who are new in biology field and microscopy field, they can get some very cool microscopy tutorials from here. So this is about like, so this is the like gist of the work which we have done this year on GSoC. I believe one more thing, yeah. So this is the use case diagram, which I guess every contributor who are coming to contribute in the DevoWorm AI project has to keep in mind. Like this is the main architecture that we have made it. Like this is the basic site map, if you will, of which you have to follow when you are making new contributions here. So there are like different things that different pages which are listed here. Can you see? Like, can you read it?
That's good. Yeah.
So there are different pages, and like you can add more and more things here. But this is the basic sitemap which we are going to follow. And like, if you are trying to like adjust it or like manipulate the basic sitemap, you have to be a very strong reason about it because whole architecture is stand up on this UML diagram, which we have given a lot of time and we have given a lot of suggestion on it, and after that we have finalized it. So this is the last, I think, closing comment from my side. And if I can, so that's the this year's GSoC journey. I have a wonderful journey with all the people here. Like we got things are very really smooth here. Like I even got sick for about 8 to 9 days between, but still we are able to complete a lot of things. So if you have like any questions, you can ask.
Yes. Oh. Well, thank you, Ujjwal. That was good. Any questions? I had a couple comments. So the picture you showed, the UML diagram? Or did you bring that back up? Or yeah, that one. So yeah, you could maybe put this in the location, where we have contributions. I don't know if we have a contributors tab on in the organization,
Yeah,
but yeah, we should have like a
yeah, yeah.
place.
Yeah, there is right now in the import DevoCell integration, like this repo is not updated because like it is not a primary repo where we have made. But yes, I will definitely put it into a contribution email, and I have also put it on the document that I am preparing for the new contributors, which it is almost complete. Like as I'm trying to make it as detailed as I can, so that contributor has never like even have to reach me for getting something. He get everything there if he like read it. So I'm trying to make it, and I will be like, yeah, definitely posting it there as well.
Yeah. The other thing is a C Elegans segmentation tab. Open that up. Yeah. So there's a misspelling here of C Elegans. There should be an S at the end of elegant.
Oh, yeah, yeah, sure. Like,
Yeah.
I will do right like after my presentation right now.
That's good. Okay. Thank you. And then I have a question from Susan. And well, Tom had to go. He said super presentations. Susan says, would DevoLearn be good for high school students, high school learners?
Like yeah, I think like I don't know like how advanced like biology is being taught to like high school students, like because I believe me and Mayukh have like dropped biology after our like eighth on like 10th standard. We don't have biology like since last 4 5 years. So I don't know like what kind of curriculum is there for biology in high school. But I guess yeah, surely it can be a very good starting point if these kind of things are being taught there.
Yeah. I mean, well, I guess it's, yeah. So I could Let's Talk Science know about this site. Yeah. So Let's Talk Science, I guess, is a teaching resource, isn't it? Yeah. So yeah, definitely. If you know of any groups that might be interested, you should make this public now. It's open, and if you need any links, you can contact me or Ujjwal or Mayukh about that, and just we can give you the proper links and everything. Make sure that we get the word out there. So yeah, I definitely think, though, from my perspective, I think high school students could probably use this. I definitely think if they have, and a lot of high school students nowadays have a programming background of some type, so it's something they could probably do. If you're like a AP biology student, maybe you could probably do it. Maybe even a regular biology course, maybe upper level 10th or 11th or 12th grade. You could do it. So yeah, I mean, I think that's worth exploring. I bet you there's a untapped market there. And let's see, Maniak says his presentations were amazing. Thank you. And Mayukh says, I have another thing to say regarding DevoLearn. So what was the other thing?
Yeah, so I'll have to share my screen for that.
Okay.
Yeah, I'll just share. So, can you see it?
Yeah, it's coming up.
Okay. So, is it visible now?
Yes. Yeah.
So, there's this event called Hacktoberfest, which happens every year. It's basically the whole month of October where people make pull requests in different organizations. So it's something in which everyone actively, like everyone in the open source community actively participate, because they get like free goodies and free t-shirts and stuff. So I guess this year DevoLearn should participate in Hacktoberfest. I could arrange for that. So if that happens, then like potential contributors could bump into this organization and then start contributing. They might start with small contributions like correcting comments and stuff, like fixing typos in the README or something like that. But that would get them going inside the org. So I guess we should participate in Hacktoberfest. It should be like in our agenda.
Yes.
Like, it's pretty like it sometimes gets organizations a lot of.
I think like participating as a DevoLearn page would be very nice. Like we do have different kind of projects there. Like we have Python notebooks project, we have a library project, we have a Heroku app and Python app project. So like people might be interested in like and it is like as we all know, like most of the people who come to Hacktoberfest just made one or two pull requests and they sometimes leave it. So but it still can publicize our library at a
Sometimes
good
they
scale.
stay, sometimes they leave. So it's kind of an interesting thing. Like people who really get interested, they just stay and they keep contributing. So yeah, we could participate in this. Yeah.
Yeah,
So that's what I.
yeah, I definitely think that would be a good thing to target. I think we did this last year with like the Devo Zoo. There was like an open data days and participated in that. And I did like Hacktoberfest in another organization, but I also kind of publicized it here, but I didn't really get any response. But it's just like GitHub. It's very they sort of celebrate it, so people are on GitHub and they're primed for Hacktoberfest, and then they come across your project, and I think you put a badge in the repo or something, and you make a point to advertise it, and people will come by, and I think
Yeah.
there's some incentive structure to it. So
I
yeah,
feel like more if you want to participate it like it is not like only participating like me and Mayukh have to like push it in our college groups and like so that more and more contributors can come. Like people are interested in doing things like most of the problem is like they don't know where to do things. So like we can do that I believe.
yeah, it's a good opportunity to interact with people if they're coming by, and you can just well, I'll show you in a minute what we had. Yeah, Jesse says he can also help with advertising that. So yeah, that'll be something too we can tie into to advertise this. Jesse says these presentations are great, and I can't wait to start building more things with them. So yeah, I think that's good. I think definitely we should be building on this. Yeah, we should be building on this, and trying to use these things like Hacktoberfest and other things to push it forward and get people involved. So, well, thank you, Mayukh and Ujjwal, for those presentations. And I'll remind you, I reminded you in Slack yesterday that I guess you have your presentations or your submissions submitted, so I don't have to remind you of that. So,
Yes.
yeah,
Yes.
yeah. Okay, so good. Yeah. So I guess that's it on your end for GSoC. Now it's my turn to do your evaluations, and again, it's like like the other ones, and so then that'll be done, and then we'll have this project, and people can make sure that they advertise it, and definitely Mayukh and Ujjwal, you should put this on your your resume or your CV and make it somewhat prominent. We were talking about this in my other meeting on Saturday, that putting together a good website or presentation of your work is good. I think essential sometimes for people to understand what's going on. So you should have like a line on your resume. It says DevoLearn, and what you did in that process, and then have some graphic or visualization associated with it. So that's definitely something that you should play up. It's like a something for your portfolio. I think you both did a great job. There's another thing too I wanted to talk about, which was like papers that we might derive out of this work. So let me share my screen. And I know we talked about some of this before. So let me go into this. We have the. So I think I talked to Mayukh about this, about submitting to the Journal of Open Source Science. And so this is the it's a journal where it's a very simple format. You describe a piece of software, open source software. You have a couple of links here. You have a repository, an archive, a review. And then we have authors, and we have this summary. So this is a grid app, an extensible finite element toolbox in Julia. This is the summary of it. So there's a summary, and then there's some a piece of code that they put in just as a demo, and then they kind of describe why it's important. So this one doesn't have subsections, but they basically go through the software, why it's important, how it works, the major features of the software, some motivation of it, which is, sometimes there's a clear motivation. Sometimes we just did it because it's a need in the community. So in this case, they had a really strong need, really strong motivation for it. And some of these papers are more like the software is more academic oriented. So there's a stronger motivation than there otherwise would be. And so that's basically the whole paper. It's like 2 or 3 pages, and then you put the references at the end. So it's something that we can do pretty quickly, but I think it's important for another way to get things out there. So Mayukh sent me a draft, and I sent him comments, and he answered the comments. So here's where we are with this. We have the summary. We have a statement of need, which is good. And then we have to work on this a bit more to flesh out maybe, it has to be a little bit longer on this, I think, but we can flesh it out a bit. I'll put some more work into this and then turn it back around to Mayukh. And then at the submission, you just go to actually they run it all of a GitHub repo. So, we'll submit it in text. I think that's how they submit their paper. So we have to convert it from, it's easier to work in Google Docs for collaboration, but we can create, I think there's a text template that they use. We can get that and put everything in and then submit it. And so that'll be a pretty easy shot for that. And then there's another opportunity here. This is a journal called Patterns. And so this is a bit more, these papers are a bit longer. And again, it's, they're basically discussions of data science or some tool that you build. And so this might be a place to put everything together, like all of the DevoLearn. DevoLearn is like a unified thing. Like, so this is not just this little piece of software, but this is the entire platform, as it were. And so this would include like, Mayukh's work and Ujjwal's work and Vinay's work from, last year. And, we can build it up into this unified platform. Now, I don't know how, I guess it's just, this paper is on deep learning, reproducibility for deep learning. So they kind of go into an introduction. They talk about some of the concepts and terminology with respect to reproducibility, how deep learning works. So it's at a little bit higher level, or a little bit, more detail than the JOS paper, but this is still something that is, I think, generally doable for something like this. I think it's self-pressed, so it's a pretty fairly prestigious journal. At least the nameplate is prestigious, so it would be a pretty good exposure to this project as well. And so, I don't know if we'd get accepted, but we could definitely prepare something for it and then see if it goes. And if it doesn't go, we can do other things with it. It's not a total loss. But I mean, last year we did the manuscript on the Basilaria, and so that became a book chapter pretty quickly after it was written. We wrote it over. We did the stuff last summer, so that was Vinay and Ujjwal and Asmit and a couple of other people in the group. and we submitted that to we put that on the bioarchive in, I think, December. And then we got the book chapter accepted in maybe like April or May. So it was, pretty quick turnaround, several months. But that's basically, and I did this with my other group last summer too. So there are all these opportunities for, getting publications and putting things out there. And we'll follow up on these in subsequent meetings, but I just wanted to raise awareness of this. So let's see. Maybe we'll have time for a paper or two. I think if there are no more, well, we can ask questions about, this offline. I like the Hacktoberfest idea, and we'll keep thinking about ways to make this public. I think some of you have seen some of the tweets that I sent already about DevoLearn, so we'll just try to keep up that momentum of the blog post as well. So we'll try to keep the momentum up on that. We have a couple more messages. Okay, good. We've corrected the C. Elegans spelling. And then Jesse says I can also help with the advertisement. Very good. So I think it was last meeting and the meeting before we talked a little bit about agent-based modeling and diffusion limited aggregation and all this stuff. Actually, my other group we were also doing some things on agent-based modeling. So this has been sort of at the top of my head. And I think I put this paper in the presentation for the other group. This is a nice review on agent-based modeling. So agent-based modeling is something that is, you probably know what it is, but it's not usually talked about with that name. So agent-based modeling are things like cellular automata. The diffusion limited aggregation model that we saw a couple weeks ago is a agent-based model. And there are other examples using that platform that I showed you, NetLogo. And so some of the advantages and challenges. This is a review of this agent-based modeling applied to morphogenetic systems, and then how it's used and how it can be used. So the abstract is the complexity of morphogenesis poses a fundamental challenge to understanding the mechanisms governing the formation of biological patterns and structures. Over the past century, numerous processes have been identified as critically contributing to morphogenetic events, but the interplay between various components and aspects of pattern formation have been much harder to grasp. So they're trying to get a handle on the sort of the interactions involved in morphogenesis. So they're not really looking at causality, they're looking at sort of interactions and how those accumulate over time. So in particular, the theoretical concepts of reaction diffusion systems and positional information proposed by Alan Turing and Louis Wolpert. And so Alan Turing, as you'll see if you go to our DevoWorm curriculum, we have a bit on a little tutorial on reaction diffusion systems and Alan Turing's approach to it, the chemical morphogenesis. And then Louis Wolpert, which is the positional information, which is where, the idea that cells use their position to sort of signal to each other, what to become or their role in the embryo. And so, okay. Development is not a reaction diffusion system. Yeah, it's not like Turing proposes chemical morphogenesis. So this is I don't know I haven't gone through I don't know exactly what they're referring to in that sense, but dramatically influenced our general view of morphogenesis, although typically in isolation from one another. In recent years, agent-based modeling has been emerging as a consolidation and implementation of the two theories within a single framework. Agent-based models are unique in their ability to integrate combinations of heterogeneous processes and investigate the respective dynamics, especially in the context of spatial phenomena. In this review, we highlight the benefits and technical challenges associated with ABMs as tools for morphogenetic events or examining morphogenetic events. These models display unparalleled flexibility for studying various morphogenetic phenomena at multiple levels and have the important advantage of informing future experimental work, including the targeted engineering of tissues and organs. So let's see. So they talk about the history of morphogenetic observations. They talk about Darcy Thompson, who we talked about on growth and form, which he describes similarities between mechanical and physical systems and the shapes of organisms. Then, a decade later, Alan Turing proposed in his treatise, The Chemical Basis of Morphogenesis, a mechanistic explanation that dominated the field for several decades. And so the core concept of this theoretical explanation was the by now widely accepted RD mechanism, which under the right conditions, a two-molecule reaction system is capable of producing periodic patterning through diffusion instability. And so this is a little bit, this isn't like there are different ways you can look at the reaction diffusion model, and people have used this in terms of describing patterning. So this is like a very limited concept. Usually when you look at the way that these chemical morphogenetic systems are implemented, they're usually trying to predict patterns, like striping patterns or like the coat of a animal where they have spots, or the wing of an insect where they have different coloration patterns. And so the cells are actually achieving some sort of order through this process. And so that's what they're getting at with this. And so then the RD patterns produced by the inhibitor and activator gradients can be considered chemical pre-patterns that act as templates for future differentiation. And so the apparent initial homogeneity of an egg or a cell cluster morphs into spatially distinct profiles of an invisible set of high and low concentration regions. And so this involves some sort of chemical signaling. And so these signals then can be implemented in the agent-based model. And so you see how I think we saw in the diffusion limited aggregation models that I showed, that you can build different types of models with different initial conditions, different agents as cells or units, and then things that govern their interactions. And so those can all be specified. So agent-based models are a theoretical model, much more even than chemical morphogenesis, which is really a theoretical sort of description of what's going on in development. Is it the best theory? Well, probably not, because it's not, it's limited in terms of its scope, but we can use it as it's something that might be useful to describe some of this stuff that's going on. And then so Lewis Wolpert talked about positional information as a mechanism of pattern formation during morphogenesis. And this is different from chemical morphogenesis because you're looking at sort of local specificity. So this was inspired by old observations during the morphogenesis of sea urchins, which Hans Dreitsch had made as early as 1891. So in positional information, a cell is able to determine its assigned fate from its position relative to other parts of the organism. The position, in turn, is characterized by the concentration of a morphogen, which is a theoretical thing. It could be any sort of like signaling molecule. Thus, a cell senses its positional value by interpreting a morphogen concentration and makes a fate decision based on this local information. So it's different from the reaction diffusion in that the spatial location is very specific. The reaction diffusion model is more general in terms of like gradients and things like that. But this actually has there's no pre-pattern in the embryo in terms of so positional information makes this claim that there's no pre-pattern in the embryo. And so a reasonable biological implementation of positional information could be a morphogen source leading to a spatial morphogen gradient that gradually decreases with the distance from the source, thereby providing positional information. So they're linked concepts, but they're not. I mean, they're also competing concepts because they make different assumptions regarding the outcomes. And so this is applicable, I guess, to things that are maybe more explicitly embryonic or embryogenetic. And so also with respect to limb development, regeneration, and transplantation, this is also very useful. And so but there are some caveats. So as Wolpert himself recently stated, there is no good evidence for the quantitative aspects of any of the proposed gradients and details of how they are set up. So it's not really clear how these models are implemented in living organisms. It's just like a theory that maybe is very broadly fits the data, but it's not. Sometimes it does seem a bit far-fetched, but nevertheless, we can reduce this to mathematics. So we have mathematics that can describe these theoretical models, and then we can take these theoretical models and implement them as agent-based models, where you go beyond like mathematics, and you can say, if we use these rules, can we produce the same outcome? Can we basically solve the equations with this computational model? And so that's how your agent-based models come in, because you have the ability to simulate a highly parallel system using this basic theoretical framework. An equation, which might take a lot of time to solve, or it might be hard to generalize to a large population of cells, since you may be describing maybe two cells that are coupled or something like that, a very general relationship or a very specific relationship. With your agent-based models, you can do this in parallel, and you can simulate populations, and you can do this simultaneously. So you can get these things, you can study things like emergence, and you can use a set of parameters to sort of describe what's going on in that population. So there are different ways you can set up the ABMs. So you can use a number of different models that have been, pre-made for the user, or you can make your own. And they give, in this review, they give a couple of different physical representations of agent-based models. So you can use things like a lattice, which are a grid of cells where there's like a single cell in the middle, and then it's updating based on what the state of its neighbors. You have the cellular POTS model, which is actually something that we've discussed with respect to CompuCell 3D, which is a platform that we've tried to get working in our group, but we haven't really gotten very far on it. The lattice is more representative of a cellular automata. So we've done things with Morphozoic, for example. That's a lattice model. And then there are lattice-free models, which are actually more where they, you take a single centroid and you calculate a radius and you calculate some sort of sphere. And you use that as a way to delimit, where the cell is being influenced by. So everything around the cell, there may be a lot of signals going on around the cell. The cell has maybe a radius of influence, and within, whatever is within that radius is influencing the cell. So it's not it's not limited to just grid squares or these sort of grid points. You actually have a radius that can be expanded or contracted, and that can lead to different results. So you have different ways you can simulate these systems. And you can do different things. You can set up different types of theoretical scenarios. So you can use things like a proliferation model, a migration model, differentiation model to describe sort of morphogenesis. You can use, you can program this in different ways so that you have different things that are influencing each cell, in different ways. So you can use a grid, you can use a radius-based approach, and you can get different types of results that are really exciting. So you can get these different modes of dynamics like slow division and fast division. You can get this pattern formation over time. So, you're maybe specifying that a cell divides, it also differentiates. And then you can end up replicating things that you might see in a real embryo. And so one of the key features of agent-based models is that they specify unique growth rates for each cell type within a simulation. So you're dealing with cells that proliferate, and that's really one of the key morphogenetic factors in an agent-based model. And so in a developmental context, this variability and proliferation can occur through numerous mechanisms. But the most archetypical instance involves inherent differences in division times between cell types. And so that's one of the ways you can get like these differences in the agent-based model, or how you can trigger sort of emergence of more complex features. And so this fluidity and growth rate as a function of phenotype creates a vast potential for unique spatial organizations that form dynamically over time, even if only one only accounts for cell division and the original spatial orientation of each cell type. So this idea of proliferating cells and producing cells, different populations of cells, is key to how an ABM generates complexity and emergent complexity in particular. And so there are different ways that you can observe things like patterning. So in an agent-based model, you observe patterning and things like this, but you have to figure out ways to make that happen. And it's not really magic. It's just you have to figure out which types of rules you need to implement to get it to sort of mimic what's going on in nature. So it's a bit harder than writing an equation, although maybe it isn't. Because in an equation, you have to specify different variables and parameters and relationships, and you have to sort of hypothesize those things, and but it's harder to it's harder to really kind of see the outcome right away. With an agent-based model, you can implement these sorts of conditions, and then you can run the model and you can observe whether it fits your hypothesis or not, whether it looks like an embryo or whether it looks like something that's morphogenetic or not. And then you can just iterate it until it looks like what it's supposed to look like. And again, that can be both good and bad, because it may not actually be the way it works in the organism, but it's a good model. It's a good hypothesis. And so, let's see. And so they really get into a lot of detail here. And if you're interested in agent-based models, this is a good paper to go into in depth. We have a comparison here of 3 crypt model implementations and agent descriptions. So here they're looking at crypts where you have cells that divide or die, and it's basically they've modeled this in different ways so that it's trying to approximate cells sort of being born, proliferating, differentiating, and maybe dying under programmed death. And so you can do this in a number of ways. You can use the different ways of modeling interactions between cells and build these different structures that allow you to model cells, and then the cells run in the simulation, and then they produce outcomes that maybe they look like something that you would see in nature. And so, there are a bunch of different ways to do this. They introduced 3 ways to do this. What else? So then they also have ABMs with migration as a major morphogenetic factor. So then there are also models where they use migration as a way to simulate spatial organization and differentiation. So migration events are commonplace throughout development and pivotal for the morphogenesis of numerous tissues. The forces behind cell motility are predominantly mechanical, but the signals that trigger indirect migration can be mechanical, chemical, electrical, or all three. And so cells in vivo are experiencing a multitude of forces from the environment and neighboring cells. And expansion of a cell population can lead to passive movement in the cells in response to a set of physical interactions. And so migration can also be used in a way to facilitate this sort of morphogenetic organization. And again, it depends on the system that you're looking at. In some systems, this is much more common. In other systems, proliferation is much more common. So, if you're looking at like a model of an organ and you're looking at like tumorigenesis, one model might be much more suitable than, say, another model like embryogenesis or something else. And so, and then there's also differentiation as a necessary morphogenetic factor. So you can use differentiation as another standard for your model. So there's a lot in this paper, and I'm not going to go because we're already over time, but I just wanted to point this out. There are also they list limitations here, so there are a lot of limitations to the agent-based model approach, but I think it's a very good approach if you're interested in really how cells go from like an undifferentiated mass to this highly asymmetrical, organism. Then this might be a way to like kind of really get at some of those questions. And again, we have experience with this in the group. We've done a Morphozoic, which is something that is Tom, who was here earlier. That was his baby. So, I don't know. Maybe we can do something with that going forward. We have the CompuCell 3D, which we've still yet to really kind of capitalize on. And we have other models that we can use as well. And so these are all possibilities. This goes beyond what we've done with DevoLearn. I think that DevoLearn is a very good step forward. And I think that maybe the next step is to incorporate the data into some of these types of models. So let's see, what do we have here? Susan has a lot of comments here. All right, so development is not a reaction diffusion system. It plays a role, but is not the main driver. My view is that what I've learned from Richard Gordon, the interaction between, who's also in this group, is not here today, the interaction between the mechanical and chemical aspects of the cell and is not diffusion. So, yeah, development is a reaction of a living cell to mechanical and chemical responses of the cell. And then she says cells cannot think. And then there are physical links between the force of the cell, that the force of the cell experiences in nucleus. There is a physical and chemical cascade that will change the DNA in the nucleus depending on the forces of the cell experiences. Richard Gordon needs to be here and add to what I am saying. Yeah. I mean, we can talk about this in future meetings. And so then, yes, the migration explanation is more like what happens. So yeah, I think that's those are good. Those are some pretty good observations. Thank you, Susan. Again, like we can talk more about this paper in future meetings. It's a pretty long one, so but that's like that's a way forward. I think we can see maybe that'll yield something interesting. And again, if we're talking about one last thing before we go, let's try to think about like maybe like a contribution board in DevoLearn itself. I don't know, but I mean, I know we have the contribution board in DevoWorm, but maybe we should do one in DevoLearn as well to maybe facilitate some contributions there. So,
yeah, I was going to ask about that because I'm looking for ways to specifically what to do and kind of seeing a list of. I know there are issues and things like that, but however, how is best to do that? I'm curious about.
Yeah, I mean I think if there's something, we'll try to think about ways forward. This is why I presented the agent-based modeling stuff. If you want to contribute something very specific, we can talk about it. But we should also probably have a board. I don't know if we should have a board separate in DevoLearn or if we should have in DevoWorm or what. I think if you're talking about people who are casually engaging with DevoLearn, then we should have a board there. But and then we can like put things in there, and people can add things into that board. If you want to be a member of DevoLearn, you can join. I can make you a member, and then we can then that way you can add to the board and do things like that. I don't know exactly what to say about like contributions, specific contributions right now. I think what we need is maybe like some publicity for this, maybe some thinking about how we can go from like what we have in DevoLearn, which is largely data analysis and algorithms that analyze data, to like what do we do with the data? Can we do like a theoretical approach to the data and so forth? I think that's one way, because it's like we can propose something really grandiose, but it's kind of like how do we get there? And so I think, or something very, very, there's some, I think Mayukh had a beginner issue over the weekend where there was something that was code related that could be fixed. And so those are sorts of things that we might do.
Like right now, there is an issue which is like going on. Like we have some problem in importing connections from ABC library, which I believe like one of the trick to do things right is just to try and accept like there is some version problem in that library, so you can just try and like when I try to import the library, if it's not importing or it is showing error, you can go to accept and you can import the newer version of it. So this is just a roundabout and it is not a complete solution. So like I think it might be a very good first issue to like work on and like.
Okay. Yeah. So if you have issues you want to see, you can either put them in Slack or put them on, make a board in well, we'll probably just make a board in DevoLearn and put them there. But we'll try to organize it sometime soon, the next couple weeks, to get everything like going. And then you can advertise different issues too. You can say this issue really needs to be addressed. I have done that before in the meeting emails, which makes it kind of convenient for people to kind of focus in on one thing.
Like this year, like me and Mayukh and Jesse, even like we can get in touch with like in the Hacktoberfest, the main contributors are like the new students who are like in their maybe first year or second year. So like they are focused college groups like clubs which are focused to onboard them on open source. Like in my college itself, there is a club known as Build, whose former head was Siddharth Yadav, and it is basically focused on like onboarding more and more new students on open source. So like we can advertise our DevoLearn repo there because like lots of students are like engaged in this stuff.
Yeah, definitely. There's a lot of excitement around machine learning, so that should be like a hook.
Yeah, and like obviously like, there's a lot of ties to the Neuromatch Academy crowd too. So
yeah. Yeah, so if we have any ideas about that, we'll discuss it more in Slack. So anyways, Susan has a question here. Axolotl embryo images need to be manipulated to show on a 3D sphere. Yeah. So this is something else that I've been thinking about, is the axolotl images. So I haven't forgotten about that, Susan. But I have we have this problem that probably isn't something that you can do with machine learning, which is to take a series of images that have been acquired from something that's rotating and put them into a spherical frame of reference. And we talked about that algorithm a couple weeks ago, but we're still kind of waiting, thinking about it. And so that might be something that we might go in, if Ujjwal, you want to look at the data, I can share with you what I have. I have some of the data, and I have a folder where I have the data and the algorithm. So I can send it to you, Ujjwal, and you can take a look at it if you want. Yeah, I'll send it to Mayukh as well. So this is something. Yeah, this would be nice. A little bit different than the other types of images we've been working with, but it's definitely something. Okay. Yeah, so that's good, and so that's good. Let's call it for this week, and we'll talk more on Slack, and see you guys, and have a good week.
Yeah. Thanks everybody. The projects are really great.
Yeah.
And look forward to working on them ahead. Take care.
All right, take care. Bye.